Hi, thanks for tuning in to a new episode number 55 of the Doctor Podcast Show. And I'm your host, Dr. Robert Seichert. As you know, I'm an ophthalmologist, and you can tell that from my eye tie today. And today we're going to have a great guest, another ophthalmologist, Dr. Eric Pierce. Dr. Pierce is an ophthalmologist in Massachusetts. He's the Chatlos Professor of Ophthalmology at Harvard Medical School, which is one of the top ophthalmology programs in the USA and the world. He's also the director of the Harvard Ophthalmology Ocular Genomics Institute, which basically studies and does research in and treats a variety of genetic disorders of the eyes. And Dr. Pierce specializes in inherited retinal disorders. Those are genetic conditions that affect the retina and cause loss of vision. So, Eric, thanks very much for taking a time from your busy schedule to come today and speak with us about a very exciting topic. We're going to talk today about gene editing to treat retinal conditions that were previously untreatable and that patients had no hope of seeing. So thanks very much for coming today.
Thanks for the invitation. It's a pleasure.
Yeah. So can you tell us how you got started and interested in ophthalmology?
Yeah, it's probably a typical story. I did a PhD in biochemistry before going to medical school and knew I
Wow.
wanted to do academic science, academic medical science. But in medical school, you have this wonderful opportunity to try different specialties on for size as you go through rotations through school. And I fell in love with ophthalmology on my ophthalmology rotation during medical school.
It's interesting. And you also did a fellowship in a subspecialty of ophthalmology, right?
I did a fellowship in pediatric ophthalmology and initially joined the faculty as a pediatric ophthalmologist at Boston Children's Hospital.
And from that, how did you get interested in inherited retinal disorders?
Kind of a mixture of forces, I guess. So as a pediatric ophthalmologist, especially after I moved to the University of Pennsylvania School of Medicine and Children's Hospital of Philadelphia, I saw a lot of kids with genetic disorders in general and genetic eye disorders in particular, and it really seemed like an unmet need. Plus, my research directed me towards understanding inherited retinal diseases. I did a postdoc studying retinal neovascularization, the kind of thing that happens in age-related macular degeneration or retinopathy of prematurity. And as part of those studies, I found a novel gene that was expressed a protein that was limited to protein expression just in the retina, and its expression was dependent on oxygen because we were studying how hypoxia caused retinal neovascular disease. But it turned out that mutations in that gene caused inherited retinal disease. I had this kind of confluence of seeing the unmet need of the kids and then discovering this novel inherited retinal disease gene that set me up for that path.
Right. So tell us about Leber's congenital amaurosis. This is specifically the condition that you're treating with this incredible, amazing new technology. So Leber was an ophthalmologist who lived in the 1800s, I believe, and he was the first to actually diagnose this condition. And congenital means it's present at birth, and amaurosis is from Greek. It means darkness, or in this case, decreased vision. How did you become interested specifically in Leber's congenital amaurosis? And can you tell us what that is? Is it a common condition? When does it start? What symptoms do patients have? How do you diagnose it? And how is it inherited, or is it a mutation?
Sure. So Leber congenital amaurosis, or severe early onset inherited retinal degeneration, is a rare genetic condition. It can be caused by mutations in about 25 different genes that are required for vision. It typically is manifest as decreased vision with nystagmus, and in this case, the amaurosis refers to the poor pupillary responses in infants. So a typical story is a parent tells their pediatrician, hey, my baby isn't responding to my facial expression.
Hmm.
Or I notice that my baby's eyes are wobbling back and forth. Traditionally, the diagnosis was clinical, reduced vision, nystagmus, and this poor pupillary response is amaurosis. More recently, the diagnosis is really genetic. There's pretty good availability, at least in the US and Europe, for genetic testing for patients with inherited retinal disorders and finding mutations in one of the known severe early onset retinal degeneration disease genes is the definitive diagnosis, usually inherited in a recessive manner.
So it's autosomal recessive, meaning both parents are carriers, and then about 25% of their children will actually get the condition. Is that how it works?
That's exactly correct. There are a couple of more rare versions of severe early onset disease that are dominant, where only one variant from a parent is required to cause disease, but those are less common.
I see. Can this also happen just by a mutation? In other words, the parents are not carriers, but the baby unfortunately has a genetic mutation in that specific gene and then gets this condition?
As recessive diseases, we usually see that when one parent is a carrier, but the other is not. And it is possible to have de novo mutations in a given gene, each born with two or three de novo variants that affect the function of a given gene. And if one of those happens to be the same gene that a parental partner has a mutation in, it can lead to disease. But that's probably about 1% of cases overall.
I see. So that's very rare.
Mhm.
Now, what's the most common gene abnormality? I know in your paper that you published, there was a specific gene that you've identified and are treating. What's the name of that gene?
So the name of the gene is CEP290, and mutations in the CEP290 gene are the most common cause of severe early onset retinal degeneration or Leber congenital amaurosis. And in particular, about two-thirds of people with CEP290 associated LCA, Leber congenital amaurosis, have one specific mutation, which is a variant in intron 26 of the CEP290 gene that inactivates the gene. It prevents the gene from functioning. And that's the target of the therapy that we've developed.
So this is a few extra DNA segments in the gene that basically inactivate the gene and prevent it from performing properly. Is that correct?
It's even more subtle than that. It's a single base pair alteration
Single base pair. Wow.
in the middle of a long intron between exons 25 and 26 of the gene that activate what's called a cryptic exon, and a part of the genomic DNA that isn't normally included in the messenger RNA that's made from the CEP290 gene, which is required to make the CEP290 protein, which is required for vision. An extra piece of DNA gets in there and prevents the message from being used.
So there's a piece of DNA there that doesn't belong, and it's just basically causing the rest of the gene to malfunction, and you can't see. Can parents be tested for this gene, the CEP gene, before they have a baby, just to make sure they're both not carriers?
If people were interested, they could be. There are carrier testing panels that are used for expecting or prospective parents. They usually focus on systemic disorders that are more common, but we are starting to see that some of those carrier testing panels do include genes or variants associated with LCA.
Yeah, that's important. I guess it's very rare, so it's not done routinely. You have to ask for it, but maybe if there's a family history of some sort of retinal or vision problems, it might be a good idea. Do you know in vitro fertilization or IVF, they test for numerous genes. Do you know if that's routinely tested in IVF or if it's not in that specific testing?
To be honest, I'm not sure. We're very fortunate to have a wonderful genetic counseling team that we work with when we see patients. And I'm sure if I turn to our senior genetic counselor, Emily Place, and ask her if she'd know right away, but I don't know offhand.
That's at Massachusetts Eye and Ear, which is part of Mass General Hospital, part of Harvard.
It's a separate hospital. It is Mass Eye and Ear, you're correct. So it's one of the main teaching hospitals of Harvard Medical School. But it's an independent hospital from Mass General, although we're now all part of the Mass General Brigham Hospital group.
I see. So I read this incredible study. You published it in the New England Journal of Medicine about 3 or 4 weeks ago, and I was amazed when I read it, the discovery that you made and the research that you did. And New England Journal of Medicine is considered the top medical journal in the USA and probably the world. So if they published it, it's really important. So tell us about what you did in this study. And first, I want to back up and talk about CRISPR technology and how you use that, and then we'll get into specifically what you did. Can you tell us what CRISPR is and also what EDIT-101 or EDIT-101 is?
Sure. So CRISPR-Cas9 gene editing is an amazing new set of technologies that allows us to manipulate the genome of a cell or an organ with facility that we've never had before. It's really incredible. The basis of the technology is the Cas9 enzyme, which can cut DNA. And what's so special about it is when it's paired up with a guide RNA, it can be directed to a very specific spot in the genome, for example, to this very specific intron 26 of the CEP290 gene. When the guide RNA brings the Cas9 enzyme to that spot, the enzyme cuts DNA at that point, and then multiple things can happen. In the case of CEP290, we actually use two guide RNAs to cut on either side of the mutation in intron 26. So we physically remove the mutation from the gene. There are a series of other CRISPR-Cas9 technologies where that general approach can be refined to make very specific changes to the genome. It's really exciting.
That's incredible. Just an analogy, it's kind of like Microsoft Word, where you have written a paragraph and then you can go in and if you decide one of the words is wrong or spelled incorrectly, you just click on it and click delete. That's basically what you're doing with this CRISPR-Cas9 technology is you're editing or removing a specific portion of DNA. Is that right?
That's exactly right. And with the refined technologies, one in particular called prime editing, which is a modification of the CRISPR-Cas9 gene editing technology, it really is like using spell check in Microsoft Word. There are a lot of steps to get that to work in patients, but one of the reasons we're so excited about the brilliance trial of this gene editing for CEP290 associated disease is we're hoping it helps open an era of gene editing for inherited diseases using all these different CRISPR-Cas9 technologies.
Right. So if you can find the abnormal gene in any disease, if you can edit that gene, you possibly could fix it. So tell us about EDIT 101, EDIT-101. How is that related to the CRISPR technology?
Sure. So you've got it right. It's EDIT 101. It was developed by the company Editas Medicines, who sponsored the brilliance trial of gene editing for CEP290 associated disease. I started collaborating with them about 10 years ago to identify early targets to try out this new technology, CRISPR-Cas9 gene editing. And they settled on this mutation in CEP290 as their first target. And I do need to give them great credit for developing, I think, a very effective approach to treat this disease. And what they did in Edit 101 was they created an adeno-associated virus, which is a gene therapy vector, usually called AAV, that's been used extensively to deliver therapeutic genes to the retina as well as other organs. And Editas made Edit 101 as an AAV vector that encodes the Cas9 enzyme and the two guide RNAs that are needed to cut out this mutation in intron 26. They did extensive preclinical studies to identify guide RNAs that are specific, that cut where they need to cut, and don't cut anywhere else in the genome so that they're safe. And they also did a lot of work manipulating and tinkering with this Edit 101 vector to make sure it had enough efficacy, enough efficiency to provide benefit to patients. And they really, I think, did a really amazing job.
Yeah, it's incredible. When I read your article, I was floored by the CRISPR, and then I was floored by the AAV. Let me try to summarize that and let me know if I'm correct. So you now have this Edit 101 CRISPR technology that you want to get into the cell to cut out this abnormal DNA. But the problem is, how do you get this inside a cell? So what you do is you take a virus called an adenovirus, which is commonly causes colds and gastroenteritis and things like that. You take out the DNA inside this adenovirus, and instead of the DNA being in there, you put in this Edit 101 into the virus. You then inject these virus particles under the retina, and the virus automatically goes into the retinal cells that are abnormal. And then the Edit 101 does its job in cutting the gene. Is that a good summary of how this works?
It's a great summary with one caveat,
Yeah.
which is we're not using adenovirus, which, as you say, does cause colds and other transient illnesses. We're actually using this adeno-associated virus, which is much smaller, has a more simple genome, but it's easier to manipulate as you described. And it doesn't cause illness, as far as we know. So it's a nice, safe gene delivery system to use.
So basically, the AAV or adeno-associated virus is like FedEx or UPS delivering the Edit 101 package into the cell.
That's exactly correct. And if you carry that analogy a little further, it's actually like a small FedEx or Amazon delivery truck that can only handle small packages.
Right.
The reason we couldn't use that to just deliver the CEP290 gene to the retina is the CEP290 gene is too big, doesn't fit inside the AAV delivery system. So that's why we use this alternative approach of gene editing.
Right. The other fascinating thing is the Edit 101, I believe, is derived from a Staph aureus bacteria. Is that right?
That's right. The CRISPR-Cas9 system comes from bacteria, and a number of investigators, some of whom won a Nobel Prize for this, studied how bacteria protect themselves from their invaders, which are bacteriophage. And many different bacterial types have the Cas9 system. And the one that fits best into the AAV vector is, as you say, the Staph aureus Cas9.
So that's amazing again, because you're taking a specific protein or enzyme from normally a harmful bacteria, Staph aureus, which is very nasty, causes a lot of bad infections, but you're just isolating one enzyme from that bacteria, stuffing it into the AAV, and then delivering it inside the retinal cells. Is that right?
That is. And what makes it even more amazing is we don't even have to take the enzyme from the Staph aureus bacteria. We just need to take the DNA that encodes the enzyme.
I see.
And package that up into the AAV vector. So when the AAV vectors transduce or infect retinal cells, the little piece of DNA that the little circle called an episome of DNA that the AAV delivers, takes advantage of the cellular machinery to get transcribed into RNA and then translated into protein so that it can do its job.
We're making this sound incredibly simple, but in fact, this probably took about 10 or 15 years of evolution and development for this to happen. Is that right?
It is, as I said, I started consulting with Editas, the company that sponsored the trial in 2014. So here we are 10 years later. It took 10 years to develop the therapy, do all the preclinical testing required to get approval from the FDA, do the clinical trial, and then several years to do the trial.
Wow, that's amazing, but incredible progress. Now, the article in the New England Journal of Medicine also has an accompanying video which shows how you got this into the retina. Can you describe it, how you inject this into the retina?
Sure. And although I've assisted at some of these surgeries in the past, I need to say that I did not do the surgeries for the trial. At Mount Sinai, we have a wonderful vitreoretinal surgeon, Dr. Jason Commander, who does the surgeries. And what Jason does is to do this injection is a pars plana vitrectomy. And then he uses a very specific injection cannula to inject a little bit, about 150 to 300 microliters of the Edit 101 drug under the retina, usually under the macula.
What's the thickness of that cannula? It looks like it's a 30 gauge. Is that right? Or do you know?
It's really small. There's a metal sleeve that's 25 gauge, like vitrectomy instruments. But inside that, there's a 38 gauge cannula that's used to make a very tiny hole in the retinas and allows injection of the gene editing drug under the retina. It creates a small iatrogenic detachment, but because the cannula is so small, when that's removed, the hole in the retina self-seals.
Wow, that's truly amazing as well.
Yeah,
So it's a self-sealing hole in the retina.
yeah.
Uh,
And Al McGuire at Penn, a colleague of mine from years ago, really helped pioneer that technique of doing these subretinal injections.
That's really amazing. The other thing that was amazing is additional technology called OCT, which you're doing actually during the surgery, which allows you to see exactly where that needle is and to make sure it's in the correct part of the retina. Is that right?
It is indeed right. And ophthalmologists who listen to this program will be familiar with OCT, optical coherence tomography. It's an amazing technology that allows us to view the retina in cross-section in patients. We usually use it, obviously, in the clinic, in the office, but it's been adapted to use during surgery so that we can tell exactly where this subretinal injection is taking place, as you say. And we can also monitor the size of the injection because you don't want to make that blob too big, and also make sure we're not getting fluid into the wrong spaces.
Right. Without OCT, you wouldn't be able to do this because you couldn't be sure exactly where you are in the retina.
So when Al McGuire was first developing this subretinal injection technique, he did not use intraoperative OCT.
Really?
He used to tell me it was by the feel of the cannula because the RPE and choroid have a different resistance than the neuroretina. And most of the early surgeries were done that way. But I think it's a great improvement to have intraoperative OCT because it makes the technique more accessible for more surgeons. And I think probably improves safety overall.
Right. And more reproducible as well, because
Exactly.
you can actually see where you're going. So far we have a combination of multiple highly sophisticated advanced technologies, and the combination of them allows us to do this. So tell us, in your paper that you published, how many patients did you treat with this?
There were 14 subjects who participated in this phase 1/2 trial, 12 adults and 2 children.
And what were the primary endpoints that you were looking for in this initial study?
So as a first-in-human use of CRISPR-Cas9 gene editing in vivo. The primary outcome is safety.
Safety.
When we started the trial in 2019 and 2020, just around the time the COVID pandemic hit,
Right.
the subjects who were injected were the first people to receive CRISPR-Cas9 gene editing in vivo ever, which is very exciting.
Right.
And we were thrilled to find that there's a good safety signal in this clinical trial. There were no so serious adverse events associated with the gene editing drug, Edit 101, and no dose-limiting toxicities. There were mild to moderate adverse events, as you'd expect, mostly related to the surgical procedure I've described that was required to deliver the drug.
Now, on top of the safety issue, which is critical, you want to make sure it's safe. Did the patients have improvement in their vision?
They did. The secondary endpoints were all related to trying to figure out, did editing occur in the patient's light-sensitive cells and their photoreceptor cells? And did that result in changes in their vision? And CEP290-associated retinal degeneration is a little unusual in that it has structure-function dissociation. That is, if you look at an OCT, a cross-section picture of the retina, through the macula in one of these patients, they might have light perception vision, but they still have a fairly intact layer of cone photoreceptor cells in their macula. So that led to the hope that if we could restore function to some of those cones, it would improve vision. And we did observe that. 11 of the 14 subjects either demonstrated or reported improvement on one or more of the four main efficacy evaluations we used. One was best corrected visual acuity. One was the ability to detect levels of light, or it's called full field sensitivity testing, FST. Another was the ability to perform a maze test at different levels of illumination. And the fourth was to fill out a questionnaire about their quality of life. So 11 of 14 showed or reported improvement on one of those metrics. And gratifyingly, 8 of the 14 demonstrated improvement in cone function, either by improvement in best corrected visual acuity or in being able to detect lower levels of light, especially red light, demonstrating it was mainly their cones that had improved function.
So you basically demonstrated that by removing or excising this abnormal piece of DNA, the cellular DNA is able to start producing the normal type of CEP290 gene-related protein, and then the cell starts to function, right?
That's exactly right. That's what we believe happened. I do feel that the improvements in vision that we measured, especially these improvements in cone vision, which I think are quite dramatic, demonstrate that in vivo editing took place, just as you described.
Do we know how long this lasts? In other words, you were able to fix the cell. Is that a permanent fix, or do you know if these patients will require intermittent injections every few years? Is that established yet?
It's not established yet, but the expectation is this is a permanent treatment. Ideally, it's a one-time treatment, because once the editing takes place in the DNA of the cone photoreceptor cells or other cells that are transduced, it should be permanent.
Yeah. Now, I noticed that you pre-treated the patients with steroids systemically and before the surgery and after the surgery for a while. Why were the steroids used?
It's been observed in some other studies of AAV gene therapy that occasionally treated patients get inflammation following the subretinal injection of the AAV vector. And the way that's been managed is with oral steroids, as you described. And there's a pretty good track record of pre-treating for a few days and then continuing treatment for several weeks after surgery to reduce or avoid the postoperative inflammation.
Makes sense. Now, I also noticed in the study that you checked to see if the AAV vector, the virus, was present in other body tissues after the treatment, and indeed it was. Can you tell us about that? What other tissues the AAV went to and for how long?
The AAV was detected transiently in tears, but really didn't show other sustained systemic spread. For example, one of the important safety issues is to make sure that there's no change to the germline from this editing. We're really doing somatic editing, the retinal alone. So, for example, when sperm was tested in male subjects, there was no evidence of the edit 101 vector in sperm at all.
Right. You want to make sure it doesn't get into the testicles and sperm and also the ovaries and eggs to
Exactly.
possibly affect those genes. And how long has this vector been around?
Sorry, how long has
The vector, the adeno-associated virus?
so the AAV or the adeno-associated virus vector has been used for gene therapy for coming up on 20 years.
20
For
years. Wow.
example, um, for example, the first gene therapy for inherited retinal disease which was done using an AAV vector, and that trial started in 2007 at Children's Hospital of Philadelphia. And I was fortunate to be part of that study team as well. That original gene therapy approach using an AAV vector to deliver a therapeutic gene for a different type of inherited LCA was developed by Dr. Dean Bennett from the University of Pennsylvania.
So this vector has a long safety record, long track safety record. So we're pretty sure it doesn't cause any other problems or issues.
That's correct. It's
Yeah.
been used not just for treating inherited retinal disease, but it's also now a part of approved genetic therapies for other inherited disorders like spinal motor atrophy.
That's amazing. Now, can you use the same technology to treat retinitis pigmentosa, which is another inherited retinal degeneration, which is more common than the Leber's congenital amaurosis?
Yes, we can. You know, most genes that are associated with disorders like LCA or RP retinitis pigmentosa are small enough to fit into the regular AAV vector, that small size delivery truck.
Right.
So for most types of RP, I think typical AAV gene therapy can be used, where the therapeutic gene is packaged into the AAV rather than the CRISPR-Cas9 editing reagent. That vector is injected into the retina, and a new copy, a correct copy of the defective gene gets delivered to the light-sensitive cells, the photoreceptor cells. That works very well. There's an FDA-approved drug for that already for RPE65-associated disease. So I think that would be the first choice for most inherited retinal disorders. But like CP290, if a gene's too big, like the UCA2A gene, which is the most common cause of retinitis pigmentosa, then you need an alternative approach like CRISPR-Cas9 gene editing. And we're very excited about the possibility to use gene editing to treat, for example, UCA2A-associated retinal degeneration.
Well, what was that last condition? I didn't hear it.
Sorry. So mutations in the UCA2A gene are the most common cause of retinitis pigmentosa.
I see.
They cause ocular syndrome type 2, but they also cause non-syndromic retinitis pigmentosa. That gene is very large, so typical gene therapy can't be used for that condition. But there are multiple CRISPR-Cas9 approaches that we believe can be used to treat that effectively. And we and others are working actively on developing those approaches.
That'll be incredible because I have many patients who have retinitis pigmentosa, and there's basically been no treatment for them. I do cataract surgery on them because many of them develop pretty bad cataracts, and that improves their vision a little bit, but the underlying RP or retinitis pigmentosa persists and get worse. So this is finally some new hope for many patients who previously didn't have any treatment.
We hope
Where,
so.
yeah, where's this headed, do you think? Are you doing more studies? Are you treating more patients? What will the FDA require in order to approve this treatment so that it can be used all over the country and world?
We'd like very much to pursue a phase 3 trial with the goal of getting FDA approval. I think the results of this phase 1/2 study support that. Editas Medicines, the company that sponsored the trial and, as I said, developed this drug, has decided for business reasons not to pursue ophthalmology in their portfolio further, but they're
Really?
very supportive of our seeking another commercial partner to pursue phase 3 trials with. So we're working with them towards that goal.
Hmm. There are a few other large pharmaceutical companies that do CRISPR work, I believe. So maybe
There
one,
are.
right. So,
So, we're hopeful. We hope the New England Journal publication generates additional interest and will help us find a path to a phase 3 study, because I would love to see this approved, and I think it warrants it. The FDA would want to see definitive evidence of improved vision that results in improved visual function. They don't want to know just that the light, that patient's ability to detect lower levels of light is better, but they want to know that that translates into better quality of life and better visual function for them.
right. And you've demonstrated that to some degree in the patients.
I think we've got a good start. I think we'd have a little more work to do on refining endpoints for a phase 3 study, but I think that's all very possible.
All right. Hopefully, you'll get a sponsor or a pharmaceutical company partner you can work with. Do you also get funding from private sources, foundations, other entities?
So for the overall research program in the Ocular Genomics Institute, we get funding primarily from NIH grants, but also do get funding from philanthropy, either foundations or individuals. For clinical trials, which are so expensive,
Right.
that we really, up to this point, have depended on partnerships with biotech and pharma companies.
Right. If any people in the audience are interested in donating funds or money to you, what's the best way to do that? Do they go to your website or some specific website where they can donate?
They could go to the Mass Eye Near website and find the development page, but we like to try to interact with those people and meet them, try to understand what they're interested in supporting so we can best direct their philanthropy. So they're welcome to reach out to me directly, and we can talk to them and understand what they're interested in, and then connect them with one of the Mass Eye Near development officers.
All right. I'll post your web page or website on the program so that people who are interested can contact you directly, because there are
Fabulous.
many people out there with retinal disorders, inherited, especially retinitis pigmentosa and other conditions. And I'm sure many of them would want to donate. I hope that they do.
That's
So,
very generous of you. Thank you.
yeah, that'll be great. So this has been incredibly informative for me, and I'm sure the public out there. And this is very exciting. I think it's just the beginning, but I think this is going to lead to great things in ophthalmology and various genetic and inherited disorders. And I think the results we get from this will become applicable to other inherited diseases and conditions of the body. So I want to congratulate you for doing this incredible research and work. This is truly God's work, changing the genome. It's incredible.
Well, thank you very much. We have the same aspirations. And I just want to close by saying that one group we really need to acknowledge for helping make this contribution are the subjects who participated in the trial.
Right.
And I don't think we realize how much of a pioneers that they are. I think it's almost like they're the same kind of pioneers as the first astronauts who went to the moon. right? They volunteered to participate in a first-in-human study and
Right.
really have helped move biomedical research forward. So we owe them a great debt.
Right. Very important point. Thanks very much for coming today. We really appreciate it.
Thanks very much. It's a pleasure to speak with you.